Welcome to another Biochemistry MCQ series. My name is Dr. Raymond Ibe. Today we'll be looking at other questions involving proteins, amino acids. Now let's start with the first question. Protein 1. Which bond links amino acids together? The peptide bond. The peptide bond links amino acids together. Peptide bond. So if you have the first amino acid, I'll have H, the alpha carbon, the amino group, hydrogen, let's link to this one. Then we have the arrow group that differentiates every amino acid. Must have seen this in my previous videos. So we have another one here. So we have this is NH2 hydrogen. We have arrow group. So this one, this NH2 here, will react with this one to give us water, to liberate water, and the two amino acids join together. So the bond, this bond is written as C, oxygen, N, H, then the carbon, carboxyl carbon. So this bond is called the peptide bond. Remember, this carbon is connected to the alpha carbon of the first amino acid. So you have the amino group here. So no matter how we elongate the chain, we always have a carboxy end and amino end in every protein. So the answer to that question is peptide bond. It's a bond that links amino acids together. Question number 2. Secondary structure includes, there are only one option here, which is alpha helix. The second option will be beta plated sheets. Plated sheets or beta sheets. Some people just say it's beta sheets. So the answer here to this question is alpha helix. That's the secondary structure that is found here. So the answer to that question is A. Now, beta sheet is stabilized by dash hydrogen bonds. Hydrogen bond is what stabilizes secondary structure, either alpha helix or beta sheet. It is the hydrogen bond that stabilizes this structure, these protein structures. Question 4. The protein denaturation does not break the primary structure. It does not break it. So when we talk about denaturation, not that it will just break the amino acid, no. It will just make them to lose, if they are in secondary structure, it will make them to lose the secondary structure. If they are in tertiary structure, it will make them to lose the tertiary structure, and they will not be seen as a primary structure. That primary structure to the sequence of amino acid. The sequence of amino acid. Just that's the definition of primary structure, the sequence of amino acids. So sometimes you ask about what determines the sequence of this amino acid. It is the gene that determines the sequence. So once the mutation in the gene, there might be an alteration in the sequence of the amino acid. And once there is alteration in the sequence of once there is alteration in the sequence of the amino acid, that protein would likely lose its biological function. So it is the DNA that determines the sequence of the amino acids that are found in proteins. Hemoglobin is an example of dash simple protein, no conjugated protein. The example of conjugated, why we call it a conjugated protein, because it is made up of hem and globin. Hem and what? Globin. hemoglobin. So the heme, which is Heme, it is the heme plus globin. The globin is the protein segment, while the heme is a non-protein part of the protein. So it's actually a conjugated protein. Casein is actually a protein that is usually found in milk. The milk protein. Which protein acts as an antibody? The answer is immunoglobulin. Collagen serves structural purposes. Collagen serves structural purposes. Hemoglobin serves transport purpose, but the transport purpose of hemoglobin is just for CO2 and oxygen. Oxygen and carbon dioxide. That is the function of hemoglobin, to transport these two molecules. Albumin is also a transport molecule, but not for oxygen and carbon dioxide. It can transport any other thing apart from this. So it's actually a transport molecule. It also serves to buffer the blood. It also has buffering capacity. So the answer to that question is hemoglobin. Immunoglobulin. Immunoglobulin. Keratin is actually a fibrous protein. You can look at your hair. Hair is fibrous in nature. Even your nails, fibrous proteins. Protein solubility is lowest at isoelectric points. That's where they have the lowest of their solubility. Because at isoelectric point, the protein will precipitate. The protein will precipitate. So that is why at isoelectric point, the protein, it will not be in a soluble form. So, and why this occurs is that, what is isoelectric point? Isoelectric point is that point at which the protein does not have any charge. It is neither positive nor negative. So at that pH, the protein will precipitate. So at those points, the protein is usually not soluble. Now, which test gives a violet color for protein? The answer is what? The biuret test is actually a test that used to test the presence of protein. Though the biuret reacts more on the peptide bond. So when there is a peptide bond, it acts on the peptide bond. So, and it is peptide bond that defines a protein. It is a peptide bond that defines protein. Pep. It is a peptide bond that determines the protein. Once there is a presence of a peptide bond, it is assumed there is presence of protein in that mixture or in the solution. So biuret test or biuret reagent reacts with a peptide bond. And once that occurs, it will give a violet color, which is test for positive protein content. Lowry test uses which reagent? Uses the following reagent. Using the following reagent. Following reagent. Usually you see it as following Sika2. Following Sika2 reagent. Sika2 reagent. So you see it as the following Sika2, which is actually the reagent that is used for Lowry test. Which amino acid forms a disulfide bond? A disulfide bond is formed between two cysteine residues. So try as much as possible to know the structure of the amino acids. So for cysteine, we have C, double bond oxygen minus NH3 plus hydrogen, CH2 SH. So we have two cysteine. The second one we have CnH3 hydrogen CH2 SH, then C the carboxyl group. So it is this SH and this SH that forms, when they bond together, it will liberate the hydrogen. So hydrogen will be removed as they bond, as they form the disulfide bond. Hydrogen will be removed. So we have a disulfide linkage. So this one usually occur between two cysteine that are far apart. So they usually form that to form either tertiary structure. Usually for tertiary structures. So the answer to that question is cysteine. Proteins with conjugated groups are called, of course, the answer is just the conjugated proteins. Conjugated proteins. Glycoproteins contain dash. Glyco talks about carbohydrates. So they contain carbohydrate moieties. So the answer is B. Do not forget to subscribe and click on the notification bell for more videos on biochemistry MCQ series.